All right, let's get into active transport. So, just a reminder: facilitated diffusion is considered passive transport because it's going to move solutes down a concentration gradient. It cannot go against concentration gradient, and it doesn't use energy, unlike active transport. Even though facilitated diffusion does use proteins, some transport proteins, mainly pumps, can move solutes against their concentration gradient, as you'll see in a second here. Reminder, active transport uses ATP, uses energy. And the sodium potassium pump is one type of pump, and pumps are a type of active transport system. And I would say the sodium potassium pump is probably one of the more well-known pumps. So we start out here. You can see on the inside of the cell down here, we have a lot of potassium, but not a lot of sodium. Even that being said, the cell still wants to get the sodium out of it. And a lot of times this could be used. This is how you have an action potential. The part of the reset is reset of the cell in order to get another action potential in nerves is to run the sodium potassium pumps. So it's fairly important. Anyway, you've got 3 different sites where the sodium can land on the sodium potassium pump. So once all 3 of these sites get filled up, the pump gets phosphorylated with ATP. So there's your ATP down there. All right, it donates a phosphate and turns into adenosine diphosphate. All right, what that does is it changes the 3-dimensional shape of the protein, and it kind of closes on the bottom end and opens on the top. Boink. And due to the 3-dimensional change in shape, that kind of kicks the sodium out of the pump. And then you can see on the right-hand side, we've opened up some areas where potassium can actually fill in. So potassium comes in, and once both potassiums kind of land in those sites, the phosphate will be released. And then once that phosphate kind of falls off or gets kicked out, then the protein goes to its original shape and allows the potassium inside of the cell. And then it can continue. And you can see here where it's just a continuous cycle. And remember, sodium is low on the inside, high on the outside, but we're still pushing the sodium out. Potassium's low on the outside and high on the inside, and we're bringing potassium in. Okay, so here you can kind of see how it rotates, how it goes. Okay, so that's the pump. Here you can kind of see some comparisons between passive transport and mainly just the pump here. So diffusion, you're going high to low. Facilitated diffusion, you're still going high to low, but in this case, with active transport, you can go from low concentrations to high, or at least you can move materials from low to high. So we've got this concept called bulk transport, where we're moving large molecules across the cell membrane. If they're leaving the cell, we call it exocytosis. So think that it's exiting. If they're bringing it in, it's called endocytosis. And you've seen exocytosis before when you talked about secretory vesicles and the endomembrane system. So, yeah, we're not really going to belabor that one. We're talking mainly about endocytosis. Endocytosis is kind of found in 3 different forms. We got phagocytosis, which refers to cell eating. It's eating an object. Pinocytosis, where it's drinking like fluid, if you will, and then in a special case called receptor-mediated endocytosis. So if you look here at phagocytosis, you have a cell. It finds a piece of food. It could be another cell. It just could be like a kind of a macromolecule-rich item. And what it does is it extends little arms around that food particle. All right. Keep in mind it's using the cytoskeleton to do that. So it's putting those actin filaments into the cell membrane and making those arms. If you look on the right here, you can see an amoeba, and it found bacillus bacteria, and it's surrounding with its arms, and eventually it will engulf it and then bring it in like a vesicle, and it will have food for a while. Pinocytosis, it's slightly different, but yet similar. And what those cells do is they make a little pocket. You can see here on the left, they make a little pocket, and then that brings in the fluid in the surrounding area. There might be lipids or ions or something that wants to bring in, or just kind of a whole little area. Probably not ions, but anyway. And then it just brings kind of random fluid in, and it's done. And here you can see like the lining of a blood vessel that's doing pinocytosis. Somebody looked at this originally and thought they were like little caves in the 50s, and there's a term like cavulae or something like that. Anyway, that's pinocytosis, or cell drinking. And then we have receptor-mediated endocytosis, which is very specific. So phagocytosis is fairly specific for like a cell or a piece of food. Pinocytosis is a little less specific because it's just getting stuff in a general area. But then receptor-mediated endocytosis is very, very specific. So the cell makes these receptors for specific items. Okay, those items, whatever it is, will land on those receptors. And once enough receptors have been kind of filled, it creates this little pocket here, which is kind of similar to pinocytosis instead of making these arms like phagocytosis. Anyways, it makes a little pocket and brings the materials in, gets the materials, and then it'll actually recycle those receptors so they can be used. All right, so in exo and endocytosis, keep in mind all of them are utilizing ATP. They're utilizing energy compared to our passive transport. All right, and that's it for cell membrane transport.